Remember that variation job we just submitted? We used 3 different run numbers, comparing them to a genome that I had assembled and annotated in BV-BRC that was a part of the same experiment. Well, we're going to look at those job results now. So there are several ways to navigate to your jobs results. You could go to the workspace and navigate to it, or go to My Jobs here, or go to the Jobs Monitor down here at the lower right. You can see I've run a lot of jobs, but this is the one we just ran. So let's look at it. So I highlight the row and then I click on the view icon. If it had failed, I might want to do report issue to say what happened with this job, or I might want to rerun it. I did it with a specific aligner and snip color. I may want to try different ones. So at this point, we're just going to view it. And here are all the results. Let me push this guy over. Okay. So this is the results landing page, and you will notice that there are a lot of files and a lot of things going on here. The service is amazing in the kind of things it gives you. At the top of any BV-BRC results landing page, you have this, which tells you the information about the job, the ID of the job, how long it took to run, and you can click on the parameters and it'll tell you what data you submitted in the form of read files, what genome you asked it to align to, what folder you asked it to be placed into, what name you gave it, what aligner you chose, and what snip color you chose. So that's all the information there if you need it. Now let's step through some of the files that are available here. Most of these are for download. Some of them we will see in the genome browser. Some of them we'll step through looking at them on a circular viewer in later videos. And then we're going to go do a deep dive on all the files that end in TSV, which is a tab separated value that's showing the location of those SNPs. So I'm just going to briefly mention those in this, and then the separate video will do a deep dive on it. So the BAM file is a binary alignment map, and that's just showing you how the reads are aligning to the reference genome. The BAI, or the BAM BEAI file, is a separate file that helps the BAM file be viewed in a genome browser. In the next video, we're going to step through the genome browser, but you could download the BAM. So like when I click on this, you can see possible things that you could do. You can download it. You can't view it. You can download it. And the BAI file, you could also download if you have a different genome browser that you want to view these things in. It's there for you. And that's part of the motto of BV-BRC. We want to give you everything, and you may think in as many possible ways as we can, because we are not sure what you're doing next with it, but so that you have the options to do whatever you want with it. The consensus file. Let me note that this is PE1. That's my first sample. When we submitted this, as you can see up here, there were 3. So PE1, PE2, and PE3. And you notice if I scroll down, each of these, PE1, PE2, and PE3, all have identical files associated with them. So I'm just going to show them for PE1. So this is the consensus FASTA. It's taken the reads and aligned them to the reference genome. So it's really a referenced alignment. And you could take that file and you could do something with it. Maybe annotate it. It might be fun. Maybe something to try. Okay. The annotated TSV, we're going to go over these in detail, but for each of these, PE1, PE2, PE3, we have this, and it's going to open a table that shows you the SNPs where they occur in the genes, and you can even sort on it. So we have different views of this same data that's showing you the variants. This is one I can click on. And you notice I can click on the view icon. So I do, and this is showing me the table, and we'll go over these things in detail as to what each of these different things are, but it's telling you what it's called in the reference and what it's called in the variant, and how deeply it's covered and how many of the reads in that location have that particular SNP. For some of these, you notice it doesn't, it's in an intergenic region, so you're not going to see codons called or amino acids called or even a gene called, but it's showing you those as well. It'll tell you if it's non-synonymous or a deletion or a synonymous or an insertion. And for some of these that are actually within a protein coding region, it'll show you the change in the codon and the change in the amino acid and the gene. We'll go over this in detail. This is the particular gene, and it even calls how important that SNP is. All right. This is the VCF file for that. So it's showing you what the SNPs are in that particular sample. So that is available for download. And then there's the variant calling format file. So this is just showing you where exactly the SNPs are. And we have it in a zip file, which is the GZ file. And this is one that we put into the genome browser for you so that you can see the SNPs on the browser. So we're going to do that in the next video. It's very nice. And then you have the TBI file. If one wants to look at that, that is there, and it's just describing what's in there. And then we start with the second sample. And then there's the third sample. And then we have a folder that's the text files for the circular view. We allow you to see these things in the genome browser, but also in a circular viewer for the particular genome. And we'll step through that in a later video as well. Now notice we have all their HTML, all their TSV. So these are two formats of the same thing. This all their TSV, for each of these, we saw that table that we could view for the variants in sample PE3 or PE2 or PE1. This is combining them all together. So if I were to open it shows you all of them, and then you can see how many have that particular SNP in that particular location. The HTML is a perhaps friendlier format because it doesn't push the columns in where you can't see them, but you do have to scroll for it. So let's look at that. And we can step through this briefly. It's telling you that in this particular sample, on this particular contig, at this particular position, a T was called in the reference and a G was called in the variant. And it scores it. Now the scores will vary depending upon the aligners and SNP colors that you use. But this, the variant coverage, it's telling you how many reads were in that area and what fraction of them had this SNP. So this had 100% of the 22 reads that were in that area for that one particular sample. It's non-synonymous, which means it changed in the amino acid sequence. So it went from a GTG to a GGG, and it just tells you where in the reference gene that position changed and what the amino acid change was there. This is the gene. If it had a ref-seq locus, we would include it. If it had a particular gene name like DNAK or something, we would include it. This is the function. This is the upstream gene, the downstream gene. the missense variant. So that's what it's called. It changed the amino acid sequence. So that's moderate. Now, the SNP variant has 4 categories: high, which is insertion of a stop codon or a frameshift mutation; moderate, which is just changing any individual amino acid, be it, by an insertion or a particular SNP. Modifier is when it's in a particular area or something that initiates other kinds of activity, like transcription initiators. And then low is for synonymous changes. So I like this particular view, the HTML view, because I can scroll around and look at it all and look for things that everybody shares. So if we scroll down, 3, all of them are in this one. And you can just scroll over and see what it was. And there's some more ones that are in, yes. So I want to look at these in particular. We're going to spend a lot of time on this particular gene. So all 3 of them have this particular SNP. And if I go over here, this is the unique identifier for this gene, and I can click on it, and it is going to open up the gene landing page for this gene, or we call it the feature landing page. And if you've watched any of these videos before, there's a lot of information on these. We have the overall view where you can find out all the protein families, all the identical proteins or genes. You can find out which subsystems or pathways that it's involved in. This dihyfolate reductase is a very important gene in antibiotic resistance. It's involved in trimethoprim resistance. So we're going to dig down deep into this, and it shows you the gene neighborhood. And it also gives you links to other things like the CDD, the conserved domain database, that you can look at too. All right, one more thing back on our results here, or actually it's 2 more things. Sorry, I have to scroll down. If you can't remember what each of those things you submitted are, the libs text will tell you. So you select that, click on the view icon, and it'll tell you who each of them were. And I know that this was number 96, number 145, and number 147. These are just a few of those over 200 samples that I chose to run this analysis on, although I've run them on all of them. And then finally, this is giving you all the information for each sample in the SNPs from the variation service. So let's click on the view. So it's giving you some output here. It's telling you who was run. It tells you in PE1, and it even tells you who it is. There were this many reads, so many mapped, the mean coverage, the base coverage, bases with 0 coverage, which are only 17 of all of those. So it doesn't even register as a percent to that value. Those with 10% coverage, and how many variants were called, and how many of those that were considered to be high quality. So this is a very valuable little summary, I think, of things. So we have this information. In the next video, we're going to look at the genome browser so you can see where those things are and how they look. And I'll see you there.